Microbiology has a long history, and there's been lots of significant contributions by many people. We're going to concentrate on just a few of the important discoveries that happened to get the field of microbiology started. To begin our discussions, we're going to discuss one of the most significant of those discoveries, and that's the discovery of the microscope. Zachariah Jansen is credited for being the first one to take lenses and invert them, and then to use the system that he created to start looking at his surroundings, and he's the first one to really observe microorganisms. It was the English scientist Robert Hooke who made some key observations because he started looking at other various organisms to include fungi, of which then they had these little structures. Now, when he saw these little structures, he called them cells. So he is the one who's credited with coining the term cell. It was a Dutch merchant known as Anton van Leeuwenhoek who also made some very key observations because he started looking at living microorganisms. And when he was looking at them, because they were living, they would move, and so he called them animalcules. Now, von Leeuwenhoek is, of course, the one credited for being the first to truly provide accurate descriptions and drawings of protozoa, fungi, and bacteria. Another key theory or part of making of microbiology is spontaneous generation. Now, spontaneous generation is the theory that living things can arise from non-living matter. Now, this theory was disputed by a gentleman named Francisco Redi, and in order for him to disprove spontaneous generation in the late 1600s, he came up with an experiment where he put some rotting or decaying meat into jars, and on one set of his jars he left them open, and on the other set of jars he sealed them. And he was using decaying meat because it was oftentimes the meats that were left out and the maggots would appear the next day. That's what gave people proof that spontaneous generation was actually a true thing. So he started with the decaying meats, he put them in his jars as described, and then when he came back, notice that those that were left open, he had the maggots on it. The decaying meat that was in the jars that were sealed, that were closed, and it prevented the entry of flies, so there were no maggots on it. And so he said he had disproved spontaneous generation. Well, supporters of the theory disputed this experiment. They claimed that by sealing the jars, Redi was preventing that vital force, and what they were meaning is oxygen, that vital force needed for life. It couldn't get in because of the way he had sealed his jars. So Redi repeated the experiment, and this time he added a third jar to it. And the third jar had a netting very similar to cheesecloth or gauze. And he put that over his jar, and he put the meats in the bottom, obviously, of each of them. And when he came back this time, he had very similar results. The unsealed flask had the maggots. The completely sealed flask had no maggots. But then the jar that was covered with the gauze or the netting, the meat did not have maggots either. Now, the gauze did have maggots on it because the flies were attracted to the meat smell and they landed on the gauze and laid their eggs, but the meat itself did not have the maggots. So Redi said he had disproved spontaneous generation. There was still some debate, but by the early 1700s, scientists just in general agreed that larger animals could not arise from spontaneous generation. But there was still quite a bit of debate on the origin of those microscopic animals, the animalcules as described by von Leeuwenhoek. John Needham actually was a supporter at this time of spontaneous generation, and to advance that theory, he tried doing a boiling broth experiment. So what he did is he took some beef gravy and he took some plant infusions and he would boil them. And once it reached a boil, okay, he didn't let it boil for long, but once he let it reach a boil, then he would pull it off the heat and seal it with a cork. And after a few days, he found that the broths were cloudy and contained those microscopic animals. So he, of course, said then that yes, this proves spontaneous generation is in effect for those microscopic organisms. Now, Lazaro Spallanzani still disputed the theory, and he thought that Needham didn't boil his broth long enough and he didn't seal it properly because he used corks. And corks, of course, are porous and allow air in. Now, Spallanzani's theory was that it's not spontaneous generation that's giving rise to these animals in the broth, but rather that they are coming from contaminated air, that it's things falling from the air into the broth. So he repeated Needham's experiment, and this time he boiled the broth, and he boiled it for a much longer period of time, and he experimented with various times of boiling, but he boiled it for up to an hour, and he sealed those flasks, and he sealed them properly by melting the ends of the flask together so that it was not like a cork, so it was not porous. What he found was that even after days, he had no contamination of the broth. He would have no contamination of the broth until he literally broke the seal on the flask and air could get into it, and then once air came into it, then that led to the contamination. So again, thinking that they had disproved spontaneous generation, but it still lingered. People still could not accept that it was the air that was contaminating the broth. Now, the debate continued until the work of Louis Pasteur in the late 1800s. Louis Pasteur worked in the dairy and the wine industry, and he would constantly see that the wine and the milk products, etc., would readily spoil, and they would spoil quickly. So he did experiments, and he would find that it was the bacteria that was in the wine or the dairy products that caused them to spoil. They became contaminated and then would go bad. So from his work in that area, Pasteur concluded that if bacteria could spoil wine or milk, perhaps they were the cause of human illness. So that led him to postulate that were bacteria the cause of diseases. Now, before he could work on that theory, he had to first go in and disprove spontaneous generation. Therefore, what Pasteur did is he devised a series of what's called swan-necked flasks. And as you can see from the picture here, what that means is he has a flask that has an S-shaped neck in it. So he repeated Spallanzani's experiment. He took the broth, he heated the broth for a long period of time, and then what he did was he took the neck of that broth, of that flask, and instead of sealing it like Spallanzani did, he twisted the neck and he made this swan-shaped look about it. Okay, so the end of that flask remained open. Therefore, air was able to get into it. The vital force, as the disputers were saying, it was able to still get into the flask. However, because he made it that S shape, or it had a curvature in it, anything in the air, so any bacteria, any dust, things like that were heavier than air itself, they would actually collect in that little curve, and they never actually made it to the broth. So any of the contaminants in the air never made it to the broth, and the broth remained sterile for long periods of time. And it was this experiment which finally disproved spontaneous generation. And through this experiment and through his work with the wine and dairy industry, Pasteur was able to postulate or hypothesize of the germ theory of disease. And what that hypothesis was it was that microorganisms are the causative agents of infectious diseases. Now, great theory, but unfortunately Louis Pasteur was not able to prove that himself. In fact, it was one of his competitors, the German scientist Robert Koch, who was the first to basically isolate the causative agent of a disease and prove that a disease is caused by a bacteria. And in the case of Koch, he was working on the anthrax disease, and he discovered the bacteria that he named Bacillus anthracis. So Koch, through a series of experiments, which we now call Koch's postulates, was able to prove that there was one bacterium that led to a specific disease. And we still use these experimental steps in associating a particular microbe with a disease. Okay, and those are known as Koch's postulates. Now, as the understanding of the various microbes and their relationship to causing diseases was growing this time, we also had to learn about how to transmit diseases. And so the study of epidemiology then formed. And epidemiology is the study or the source, the cause, the mode of transition of a disease. And Oliver Wendell Holmes was the first to really begin the field of epidemiology. He was an American scientist who worked in the childbirthing area, and he was the first one to really notice the spread of diseases in childbirth wards because of healthcare workers. And he was the first to really write about that association, that correlation. Ignaz Semmelweis was also noticing similar ways, and he implemented a new practice in his birthing ward, and that was hand washing. He almost lost his job over it because it was not needed as far as many were concerned, but he implemented hand washing so that the healthcare workers and practitioners were required to wash their hands between patients, and he noticed that the diseases reduced. And what he was able to deduce from that was that the source of that blood poisoning of women in the childbirth came from those attending healthcare workers. Joseph Lister was an English surgeon, and he took it a step further, and he took that germ theory and he applied it to his medical practices and started taking phenol and requiring that patients going through surgery, that the surgical sites and all of the utensils in the dressing were wiped down with phenol to keep it clean. And through his practice of doing that, he was able to show that microorganisms were responsible for surgical wound infections. So all of this has led to that field of epidemiology, and these three gentlemen have led us to that very well-known practice today of maintaining aseptic techniques in sterile conditions in the healthcare practice. So despite all the advances being made in microbiology and the relationship between microbes and diseases, we still didn't have a method out there to stop infections once those microbes entered the body. So in 1910 was the first true drug that was developed, or medicine that was found to treat an infection caused by a bacteria. And the infection itself was syphilis. And Paul Ehrlich discovered what's known as Salvarsan, which is an arsenic derivative. And by giving patients Salvarsan, it was able to treat syphilis. Now, as you can imagine, since it was an arsenic derivative, we don't use that product today. But back in that time, that was a major discovery. And then in 1928, in World War II, we discovered antibiotics, and that was from Alexander Fleming, who discovered penicillin. And he did so because of an experiment that had gone awry. He was working with various bacteria, and he got a contaminated bacterial growth plate. It got contaminated with mold. The mold was Penicillin notatum, and what he noticed, he was smart enough to look at those contaminated plates and notice that wherever the mold grew, the bacteria did not grow. And so he made the correlation that there was something that the mold was producing that inhibited the growth of that bacteria, and so that led him to the production of penicillin, and we still use that antibiotic today. Despite it all, that we've learned through the bacteria and all of the spread of diseases, it wasn't until the mid-1900s, 1940s to be exact, where we really even were able to move beyond bacteriology into another field of virology. And why? Because viruses are so small, we couldn't see them, not even with the microscopes we had at the day. So in 1940s, the electron microscope was developed, and that's what then allowed us to see the viruses. We were able to cultivate them, and they got to be introduced to the scientific world. And from there, we have now the field of virology, which is developing extremely rapidly. So the field of microbiology, as you know, it continues to grow, and it's one of the fastest growing divisions in all the biological sciences. So again, this is not everything, but it is a few of those pertinent dates and times and people who helped us in the field of microbiology.